hand pointing away, your middle finger's up, your ring and your ring and index finger are down. That's glucose. Glucose has this unique arrangement. So if you're flipping somebody off, you're also doing glucose.
Okay.
So the D up, the trans for alpha, and then this middle finger arrangement as such means we're dealing with glucose. So hopefully that seems okay. A whirlwind, I mean, I realize we just covered that in a couple of minutes, but it'll be covered in another lecture. But that's how you can recognize the linkage. Identify the D first, the trans or cis second, and then which way the hydroxyl point to figure out which sugar it is.
Okay.
But that's F and glucose. Okay, I will stop share. I will maybe stop abusing the 3-seal.
Yeah, no, that is an awesome trick. Now I know. Yeah, I'm always going to think of you as glucose. So
yeah, see, that's why I think like plant head.
Yeah,
my gosh, your head really is part of the
worked out. See, it's funny with my music background. I always thought of a paradiddle for glucose. So go look that one up, Todd.
That would mean I'd have to actually research something.
Yeah, you have to research something.
Okay. Anyway, we're overstepping our boundaries. Hey, Josh.
Hey, can you guys hear me?
Yeah. How you doing,
awesome.
Josh?
Great.
Okay.
I've got a quick question. Can you guys hear that alarm in the background?
No, I do not. I do not.
Great. You never know with these thin Berkeley Review walls in downtown Berkeley. Cool. All right. Well, we'll get started here. People might trickle in late, but let's get this party going. Thanks everyone for showing up. I know it's pretty last minute to schedule this in, but it's so important to get the biology train moving, just because we're going to have a lot of lectures to cover and a lot of material to go over, and they kind of build into each other, right? So this, particular lecture is very good for, kind of your basic review that's going to just slowly build and build and build and build and build, as we get into more, complex topics. So it's better to get this one done quickly. Okay, I'm going to transition over here. Let me know when you guys can see my screen.
Hey, Josh, I'm going to take off. Do I need to make you host or anything before I do that, so I don't like knock everyone out or something?
No, I should be good. And if not, they'll just rejoin the room. But thanks for letting me know.
Okay. Oh yeah, I have you as co-host there. Okay, cool. Oh yeah, I'm out.
It's
See everyone next week, and thanks for everyone that showed up. Actually, I'm gonna change that. Host. No, I'm gonna make you host because I am gonna. It had said end really? Instead of leave. Yeah. No. So if I'm host and you're co-host, if I end the meeting, everyone gets kicked off. So I did that once on accident, so I changed it. Everyone is now I could leave in peace. All right. See everyone later.
Nice. Great. Cool. All right. Okay, so today, again, thank you guys for, coming last minute. I know that, you're going to have a lot of lectures this week, but, it's important. And I know right now is, especially in the first couple of weeks of the class, I know it's, incredibly, overwhelming, right? You're probably looking at that assignment list and like, how the heck are we going to get through this? And just trust me that you'll slowly, as you start to take big chunks out of, all the homework assignments and all the readings, everything is actually going to become much, much more efficient. It's just that, initial growing pains, that are very stressful. But, honestly, that's just the nature of the beast. Let's begin the lecture. The instructor who was supposed to do this lecture, I'm actually, starting to get a little nervous. She was, a former student. She was very excited and gung-ho, about, doing these lectures. She'd been teaching in the past, and kind of just dropped off the planet, which is never a good sign, which is, a little scary. So hopefully we hear back from her. But in the meantime, we're just going to prepare, for me to do the lectures, which is no big deal. Okay. So today, talking about structure and function, this is kind of where all, biochemistry starts, right? If you open up any textbook, if you open up, any, traditional biochemistry class, the very first part is going to go over, structure and function. right? Cellular structure and function. We're going to touch a little bit on viruses. Okay, but before we do that, here's pretty much the entire roadmap, at least for your biochemistry / molecular cell biology lectures. Now, things here are going to, move around maybe a little bit. Nothing is, set in stone, but this is pretty much, the topics that everything is going to cover, right? So as you can tell, this is, an incredible amount of information. So, when you come to class, take notes, good notes, ask good questions, come to office hours if you ever get stuck or need help on any of these. Now, for this class, we are going to go over amino acids and proteins again. I know that you had an amino acids and proteins lecture, but that had more of an organic chemistry focus. And so today we're going to go for more of a, biochemistry, structure and function focus. And, amino acids are, the single most tested subject on the MCAT. So someone asked if there's any handouts for this lecture. Yeah, there's actually several. They're all in the class portal, right? You'll have like a lecture outline, and then you're going to have some supplemental readings that I'm going to talk about in a bit. Okay. But like I was saying, the 20 standard amino acids, these are hands down the absolute most tested, subject on the MCAT. Okay. And I'm sure that you've seen these figures before. This is the Creative Commons Wikipedia, chart for amino acids. If you don't already have this, I recommend just downloading it, just Google search and just having that next to you at all times. Okay. Ideally, you're going to get to know every single thing that you see on this chart. And that's going to come from either directly memorizing or, more importantly, doing a lot of practice questions and just slowly figuring it out. Okay. So that's my recommendation for that. And so when it comes down to the 20 standard amino acids, I mean, when I say you need to know them, I mean, you need to absolutely know them, right? You're going to need to know the full name, right? You're going to absolutely need to know the 3-letter code. You're going to absolutely need to know the 1-letter code. You're going to need to know the chirality, right? You're going to need to know the structure of each R group. Okay. You're going to need to know even the pKa of the relevant R groups. All right. You're going to need to know the charge, polarity, hydrophobicity, right? You're going to need to know amino acids with special cases. All right. And then, tying everything together, right? You're going to need to know the importance of each amino acid, right? Given any particular situation. And that's what's going to be tested, on your MCAT. Okay. And so if you can do this, and this is going to happen, over this course and, during your studies, you're just going to be able to get a lot of free points on the MCAT, just because there's going to be a lot of questions that come up and you'll be able to answer them. And the more you know these, the quicker you're going to answer those questions and the more time you're going to have for other questions. And so someone says, how do you recommend memorizing structure, right? And comparing structures? I mean, I really recommend just drawing them out. I mean, that's what a lot of people do. Just drawing them out, right? Oops, does this work? Oh yeah, there it is. Right. I really, just draw them out again. The good news is you're just going to get so many practice questions with amino acids in them. You're just going to start to kind of understand it as you go. Right. I would say right now, what you should do is memorize the 1-letter code and the 3-letter code. And then, as you go forward and as you do questions, you're going to start to, just by needing to, you're going to know the structures of them, right? You're going to know the pKa group, pKas of them, right? And then most importantly, you're going to know, why each thing is relevant given a certain situation. All right. So that's my little spiel on amino acids. And we'll go over, just some stuff more of a, this is a little more OChemy, but it's also, biochemy, right? The alpha carbon, right, of all 20 amino acids, right, this one, except for glycine, is asymmetric. Okay, so what that means is that the alpha carbon is therefore a chiral center. Okay, And just a quick review, right? Chiral centers, right? They have, 4 different functional groups. Right? So 1, 2, 3, 4, right? And there's 2 different arrangements possible. So this is the classic, here's a mirror, right? And you have your left-handed and right-handed non-superimposable mirror image, right? In which case they would be technically enantiomers, right? Which is a special case of a stereoisomer, okay? Oh, yeah, that's good, Alexander, yeah, right? Good comment. Okay, and then remember, molecules with chiral centers, right? They're optically active, right? Which, means that they can rotate polarized light. And you're going to go over that in lectures and stuff, when you go to light and optics and things like that. So we don't need to cover that right now. Okay. But do you guys ever wonder why amino acids and sugars are named with the L and the D when there's also the S and the R, right? Does anybody know why they do that? Why that's like that? Besides just to confuse the hell out of everybody who's ever taken a biochem class. Yeah, exactly. It's super frustrating. And, it's one of those things, once you realize, why they do that, it makes so much sense. Yes, because biochemists want to be different, right? So when you're naming something, right, S and L pretty much mean the same thing. There's a little bit of a technicality, right? And that's what I want to talk about. You can imagine, right, a glucose molecule, right? So if we were to draw the Fisher projection of a glucose molecule, right, CH2OH, right, you got your, Todd just went over this, that cool trick to memorize the glucose. I'm not going to go over it now, but you guys will go over it later. Okay, imagine trying to name this molecule, right? If you're going to use the absolute configuration, which is what you mean when they say S or R, you would have to do the absolute configuration for every single one of these chiral centers, right? And so biochemists said, absolutely not going to have to do that, right? We're talking about amino acids, we're talking about sugars all the time, and they all have their own specific name, right? Alanine, cysteine, histidine, whatever it is, they have a specific name. So why do we need to, write out this entire, R1, S2, and down the line, you can picture it, when all these things that we use every day already have a name, right? So they, created something called the reference naming system, which is when you see L and D. And it's in reference to the simplest 3-carbon sugar, which is glyceraldehyde. Okay. And so when you see L or D, it's naming it L or D based on the reference, which is L-glyceraldehyde, which is right here, right? And so if the stereoisomer is related to L-glyceraldehyde, it's termed L. And if the stereoisomer is related to D-glyceraldehyde, it's termed D, right? So if on the reference carbon, which is the second carbon, right? Here's the anomeric carbon of a sugar. Here's the reference carbon. So for the reference carbon, which is carbon 2, if the functional group is on the left, right, then it's going to be L. And if the functional group is on the right, then it's going to be D, right? And so if you wanted to name the full thing, right, S, 2,3-dihydroxypropanol, right? Biochemists got tired of doing this. So you can just do L and D to know what you're talking about. Okay, does that make sense? Right? And you guys might have gone over this. This is more OChem, right? The absolute configurations rules, right? So if you wanted to get S or R, right, you would rank all 4 of the substituents based on their atomic number. Okay, right? So this would be 1, 2, 3, and 4, right? And then if they proceed from highest to lowest in a counterclockwise, then it's S for sinister, right? And, the other way clockwise, it's going to be R, okay? So that's just kind of, where biochemists sit in terms of L&D and RNS, right? Just making your life simpler. Okay. Okay, do you guys know which molecule this is? Yeah, simpler. I know. Just wait till we get into like genes and proteins and how the naming for those go. If you want to start complaining about simple names, right? It gets totally out of hand. And that's why we just have to practice and, do a lot of these questions. Do you guys know what amino acid this is? Right? You guys should know your amino acids. Alanine, sweet. +1 point to Eric. Okay. Yeah, it's L-alanine, right? Because on the left side, and this would then be D-alanine. Okay. So let's do a little bit of trivia, right? Just while we're on it. I like trivia. All of the L amino acids from proteins are S amino acids, except for, does anybody know? Nice. So there's actually 2 of them, right? This would be glycine. It's because it's asymmetrical, so you actually can't assign it. But there's one other one, right? And this is just fun biochemistry bar trivia. It's actually cysteine, right? And why is that? Anybody? That's because if you were to draw cysteine out, right? This is actually got a higher oops, sorry, that's not there. The sulfur on it, right, is actually got a higher, oops, I erased it, higher atomic number than the nitrogen. And so it actually ranks differently. And so you still get an L amino acid, but its absolute configuration is actually the opposite, right? It's an R absolute configuration. Cool. Okay. So let's move on to pKa's. And pKa's, you're just going to, before you know it, just have dealt with pKa's so much that everything is going to, it's going to become easy. But, now we need to learn it, right? We need to start to know this because you're going to start to have to do a lot of questions that have to do with pKa. Okay. And so when we talk about pKa, right, it's good to understand where it comes from, right? So in general, for, any weak acid, right, HA, right, you can write the dissociation, HA is in equilibrium, right, with, the proton and the conjugate acid, right? Okay. And so the acid dissociation constant, right, Ka, right, you can simply write as the concentration of hydrogen over the A minus over HA, right? And so, to actually get these numbers, right, to actually get the ionization, right, of the weak acid or base must be determined experimentally. So you actually have to run experiments to collect the data, to get these numbers, okay? And when you do that, you're going to notice that these ionization constants, right, Ka is just the ionization constant, they're very small, right? So typically when we're talking, in a biological system, it's going to be somewhere between 10 to the 2 to 10 to the negative 10 to the negative 2 to 10 to the negative 13. Okay, right? And so what this means is the larger the ionization constant, right, the greater the tendency it is to dissociate. Okay, the stronger the acid. So you can picture this number is going to be, bigger, right? then the greater the tendency is to dissociate of more of these. All right. And so here's an example of two very common biologically relevant molecules. Okay, you have lactic acid, right, which we're actually going to talk about in the next lecture when we talk about glycolysis, and then bicarbonate, which we're actually going to talk about later when we're talking about blood, right, and hemoglobin and sequestering CO2. Okay, but just for reference, right, it's always good to have reference numbers. These are the ionization constants for those two species. All right. And so when you look at these numbers, you know, it's, if you were to just talk about these with everyday life, or, you know, look at these in papers or whatever it might be, it's really difficult to compare. Our brains simply just don't work well when we're dealing with, you know, 10 to the negative 11 or 10 to the negative 4. Okay. So if we wanted to compare them, we simply switch them over to a logarithmic notation, okay, which is denoted by P. And so whenever you see P, right, all it means is negative logarithm of. Okay, so you see pH, right, negative logarithm of H, right? And when you see pKa, it's negative logarithm of the ionization constant, Ka. All right, so you can write it now, pKa equals negative log of Ka. Okay, and so similarly to what we said before, right, the smaller the pKa, right, the greater the tendency it is to dissociate, so the stronger the acid. Okay, so again, the smaller the pKa, the greater the tendency to dissociate, the stronger the acid. Okay, and so as you can see, it's much easier to tell, right, that lactic acid is a stronger acid than bicarbonate because the pKa values are smaller, right? So you see how much easier it is now to compare? Cool. All right. Now, for each of the 20 standard amino acids, right, each of them are going to have their, you know, unique set of pKa values, right? You're going to have the pKa for the amino side, right? And then you're going to have pKa for the carboxylic acid side, and then you're going to have the pKa for the R group, okay? And so for your MCAT studies and just for, you know, in general, right, you can pretty much say that the pKa, right, for the carboxylic acid side is going to be around 2. And then the pKa for the amino side is going to be around 9. Okay. And then as you will find out as you're studying, right, the pKa for the side chains are going to depend on the amino acid and the situation. All right. Okay. So let's go through a quick example here. Let's take the amino acid histidine, right, which is a very, very, very special amino acid, which you're going to realize, right? And it's special in the fact that it has three dissociable proteins within the H2O window, right? So let's see here. Let's say that over here we have, you know, pH equals around 1. And then let's say over here we have like pH equals around 10, right? So if we incrementally add base, right, if this amino acid is in solution, okay, to the originally fully protonated histidine, right, as we go this way, right, we're going to remove the carboxyl, right, the imidazole, right, which is this one, and the amino protons respectively. Okay. So over on this side, right, what is the charge of this species, number 1? Okay, you've got a plus, right, and then nothing. So you have a charge of +2. Okay, now as we add a little bit more base, right, and you go look at species 2, right, you have a plus, right, a minus, right? So we removed this hydrogen here and a plus. So we have +1. Furthermore, we keep adding a little bit of base. What's going to happen? We're going to remove now this proton right here, right? And so now we're going to have a plus, a minus, and nothing, which cancel each other out. So the charge is going to be 0, right? And then here you can see that the charge is going to be just -1. Okay. So now just notice and keep this as a mental note that right in the physiological pH, right? It's around, what is it? Physiological pH is around, 7.4, something like that. Right? So around that pH, you're going to have a histidine residue Oh, pithing, sorry. This is supposed to say within. It's a typo. So histidine has 3 amino acids that has 3 dissociable proteins, right, within the H2O window. And this is not supposed to say proteins, it's supposed to say protons. That makes way more sense. Thanks for catching that. Does that ever make sense, everybody? Oh, and spell check totally. Oh, man. Cool. So histidine has an amino acid that has 3 dissociable protons within the H2O window. Okay. So back down to here, right? Notice that at physiological pH of around 7 to 7.4, you're going to have a histidine residue that is somewhere between being slightly positively charged and slightly negatively charged. Okay. So we're going to come back to this later in the next lecture, talking about enzyme mechanisms and their mechanisms of action, and how histidine plays such a pivotal role. Okay. But first, let's, someone asked a question. So someone asked a very good question. They say, why do we care about the mechanism behind this, right? So why do we care if we lose hydrogen, right? Why do we keep losing a hydrogen at different pKa values? Okay, so why we care about this, and this is going to be explained, especially when we talk about enzyme kinetics, enzyme mechanisms, is that depending on the charge of these residues, it'll interact and cleave bonds. It'll bind to different, molecular species, right? So depending on the charges of not only this amino acid residue, but the surrounding amino acid residues, they're going to play off of each other, and they're going to cause, enzymes to do their magic, right? Break structures, build structures. So, the pH of your solution is so vital, and the protonation state of your proteins and your peptides and your residues is so vital. Does that answer your question? And this is all going to become very clear as, as we go forward. But right now, it's important to know that amino acid residues, depending on the pH, will gain or lose protons, right? So they will have effectively higher and lower charges. All right.
So that was my question. I meant more so from the end of, would they really ask us which proton we lose in which steps? Can they give us figure 2 and be which is the next?
They would absolutely ask that. And that'll become second nature. I mean, I would think that'd be a very easy and in fact, they've definitely asked that question before. And it's a very easy question. You just have to remember the pKa values, right, of each of these species. So if you know the pKa value of histidine and you do your thing and you know that it's around 6.0, right, of the God, I always have the imidazole ring, right? This is this thing, right? That has a pKa value of around 6.0. You know that the carboxyl side has a pKa value of around 2.0 and the amine side has a pKa value of 9.0. Then you should know which ones will come off first and last, right? You could actually get the order. So very good question. Okay. Yeah. And they'll ask this not only for a single amino acid, they're actually going to ask this more likely with an entire peptide chain, right? So if you have, N terminus to C terminus, and you have all these residues, right? These are amino acid residues linked to each other, which we're going to start talking about later. So if this doesn't make sense to you, don't worry about it. And you'll actually have to know the total charge of the peptide based on the pH and based on the pKa values. Okay, right? And so, this is, yeah, exactly. So we'll move on, and hopefully, this will start to become, and see, someone who may or may not have taken the MCAT has been asked this, right? So, this it seems very hard and abstract at first, but the more examples you do and the more you work with it, this is going to become second nature. All right. So, what if we were to graph this, right? So what if we were to do like a titration curve, right? So just moving from a low pH to a high pH by adding base, right? What were we to do if we were to graph this? Okay. All right. So you'll notice that these species here, right? 1, right, will form a very nice titration curve all the way up to species 4 here. Okay. Now, the big takeaway here is that you'll notice that at the pKa, right? So I'm gonna write this down. When the pKa equals the pH of the solution, so here's pH 2, this is pKa 2, right? That's at half the equilibrium point between these two species. All right. So in other words, right, there's going to be equal parts of 1 is going to equal 2 at pH of 2 if the pKa is 2. Okay. So do you see that here? I'm going to slow down here for a sec, right? So when the pKa is equal to the pH of the solution, right? What that means is that you're going to have equal concentrations of this species and this species, whichever ones are on either side of that pKa value given here, pKa 2.0. Okay. And so why is this important? This is important for when we're calculating the isoelectric point. All right. So again, the Zwitter ion, right? This is the molecule that has a net charge of 0, right? So this is, if you're talking about a peptide, when the peptide's net charge is 0, if we're talking about histidine, in this example, it's when histidine's net charge is 0, which was species 3, right? So that would be the Zwitter ion in this case. Okay. So if you want to calculate the isoelectric point, okay, you take the pKa values on either side of the Zwitter ion, right? So we just went over it. 3 is the one where the charge of histidine is equal to 0. And so you take the pKa values on either side, which are given to you right here and here, right? pKa 6 and pKa 9, right? You add them together and then you divide them by 2. And that gives you this number down here, which is the isoelectric point. Okay, so this is the pH where that residue, or if we're talking about a peptide, that whole peptide is going to have a net charge of equal to 0. Okay. Oh, that is the half equivalence point, right? So when pKa equals pH, right? So we can look at it right down here, right? When pKa is equal to the pH, that means that half of your molecules are going to be in this state, in this case, fully protonated, and half your molecules are going to be protonated. So they're going to lose that one hydrogen, right? And so you'll have an equal concentration of them. Now, over here, right, if you keep adding base, right? Oops, can you guys see? There it goes. Right around here, this is where pretty much all of your molecules are going to be in this state, right? So it'll have only lost 1 proton, okay? And someone says, I thought we used pKa2 and pKa3. Okay, so try not to do pKa2 and pKa3. You have to do the pKa's. This might be confusing, this one right here. You do the pKa's that are on either side of the Zwitter ion here, okay? All right. Does that make sense? Because the Zwitter ion is when the molecule is at a net zero charge. So you take the pKa's that surround that, and that'll get you to the isoelectric point. Because everything outside of that range is either going to be fully protonated or fully deprotonated. Okay. Yeah, and Melody just brought up a shortcut. Yeah, I mean, this is something that you could look up, later, little shortcuts to doing this, whatever works best for you, doing these questions. But I think it's important to understand, what's going on, right? You're taking a molecule, in this case, it's just the histidine residue, and you're slowly deprotonating it, right, as you add more base, right? And at some point, right here, it's going to have no charge. And that pH is going to correspond to the isoelectric point, right? The PI. And someone says, could you repeat what's going on at 2 when the slope levels out? Okay, so right here, right? So let's go back to this one real quick. Let me erase this. This has everything in one slide. This is much better. Okay, so 2, this little 2 right here corresponds to this molecule species right here. Okay, and so this 2 on the graph, which is, I think it's closer to pH of 4. I don't actually know what the pH is, right? That means that pretty much all of the molecules are in this state at a pH of, if it's 4, around 4, whatever it is, okay? At a pH of 2, given by the pKa, that means that we have pretty much half the molecules fully protonated, and then half the molecules missing that one proton, right, on the carboxyl. Does that make sense, Leandro? Awesome, great. Yeah, this is, very important stuff. So if you have any questions, do ask, because, you're going to get questions, and this is all going to become second nature. So it's good to have a good foundation on this, just because all your biochemistry questions, it's almost like every single passage, you're going to have one question on this, just because they make really good multiple choice questions, right? Cool. Okay. Does anybody have any more questions on this? Okay, then someone asked, so the best buffering regions are around the different pKa's. Exactly, right? So this is something that you're going to go over in your acids, bases, buffers, titrations. So I don't want to totally botch this. But yes, these are going to be your buffering regions, right? And that's when you have equal or closer to equal concentrations of each species, right? They'll act as a buffer. Oops. Okay. Great. Now, moving on, right? Let's talk about the MCAT, right? How are amino acids tested on the MCAT? Okay, let's go over some, easy question types, some medium questions, and some difficult questions, right? The easy questions that are just absolutely free points. If you get these wrong in the MCAT, I will be very upset. Just identifying the amino acid based on its structure, right? This will be the easiest question. They come up and they're just literally free points. Just collect them while you can. Okay, a medium type of question on the MCAT might be something like, determining how a peptide, right, or a protein behaves, based on its amino acid sequence. Okay, right. So these questions require, an understanding of the amino acid side chain characteristics, right? So good examples would be, running how proteins will run on a gel, right? How different proteins will run on a gel, right? How a protein is shaped, based on its amino acid sequence, or even how a protein will elute when it's purified, right? And we'll go over this, in biochemistry lab techniques lecture and before that, and just kind of as you go, right? You're going to do questions and you're going to start to figure all this stuff out. Okay, now let's look at this. This is big, right? Difficult questions. Okay, this is the stuff that kind of needs you to have an understanding of biology, like pretty in-depth understanding of biology, molecular biology, biochemistry, whatever it is, right? Because you're going to get questions that are pretty much going to ask you to predict, what's going to happen to, whatever cellular process is being talked about. If researchers, introduce a mutation, or maybe there's just a species that has a mutation, right? Right. So change the amino acid sequence. What's going to happen, to the function of that cell or that mechanism or system, whatever is being talked about. Okay. Right. So again, would a mutation cause, a critical cellular process to be disrupted, right? So like, you might get a passage on like sickle cell or cystic fibrosis, and you're playing with different amino acid mutations and how that's going to affect the whole system as a whole. Okay. And so that's going to understand, you're going to have a very deep understanding of amino acids, proteins, the structure, and swapping things out happens. Okay. Cool. Anybody have questions on that? And this will be just best done by, going through practice questions. All right, let's move on to proteins. Okay. So, generally speaking, this might seem obvious, but it's good to just kind of let's align ourselves on protein nomenclature. Okay. So a peptide, that's anything with 2 or more amino acids, right? An oligopeptide, if you see this in a passage, right, that's anything with about 10 to 30 amino acids. These numbers are not exact, by the way, except for 2. Polypeptides, right? Same thing as a protein. These are just the longer, continuous, unbranched peptide chains. All right. Okay, and it's considered a protein when it's, again, rough, right? Not exact, about 30 or more amino acids. Okay. Does anybody know what the biggest protein is and how big it is, how many amino acid residues it is? Just another fun fact. Biochem trivia. It's this protein called titin. Okay, and it's the biggest protein. It's found in muscles, right? And it's going to equal about, I think it's 27,000 amino acid residues, which is absolutely massive. Okay. And, it contributes to the elasticity of your muscles. Okay, someone asked a good question. What the difference between an oligopeptide and a polypeptide is, right? So an oligopeptide is a polypeptide, right? But an oligopeptide specifically means a shorter chained polypeptide, right? So if you ever see, the word oligopeptide, don't think, titin or anything that's enormous. Think, somewhere around 10 to 30 amino acids, okay? If you see polypeptides, they could be referring to an oligopeptide, or they could be referring to a protein, which is usually about 30 or more, residues. Again, these numbers aren't exact. There's no like official cutoff line. You can start calling things like an actual protein. Okay. Oh, that's muscles, like your like arm muscles. Cool. Great. Yeah, good questions. Okay, so let's talk about protein structure. Okay, so when we're referring to primary structure of titin, I don't know exactly, but I think it has to do with the elasticity of your muscles, right? So allowing them to, to stretch. You can imagine like a huge amino acid chain that like you can stretch it so it doesn't just snap. That's my guess. You could read into it. It's not, not necessary, just fun facts of the day. Okay. Okay, so protein primary structure. Okay, this is just the sequence of amino acids connected by peptide bonds. Okay, that's all the primary structure is. Right, so if you have a protein or a peptide or whatever it is, the primary structure of it is just the residues connected by peptide bonds. Okay. So what is this? If you guys has anybody done their water amino acid codes? Does anybody know what this says? Yep, that's what we're going to do. This is for now on our favorite peptide. Now, I might ask you, what's the charge at pH of 7.5 at physiological pH? We're not going to do that right now, but that would be a question where you would have to go through the R groups, the pKa's, and know that this has got a 9.0 and this has got a 2.0 pKa, right? I know this has got, I don't know, 12, 13, I forget, right? And to go through all those and actually get the net charge of this peptide. Totally valid question. Okay. Okay, so if we're going to talk about primary structure, right? If we're going to make a peptide, what needs to happen? So here we have two residues, right? We got amino acid 1, we don't know what it is, and here's got amino acid 2, right? How do you get these things to form a peptide bond? Okay. You're going to need to do something called a dehydration synthesis, right? Which is essentially dehydrating it, right? So it's taking out the water, all right? And what you're left with is now not two single amino acids, but a peptide, okay? And so again, if you ever hear the N terminus or C terminus, N terminus is the amino side, and then the C terminus is the carboxyl side, okay? And so if, again, these are free points on the MCAT. If they ever ask a question, they say, point to the peptide bond. You're going to point to right here, and you're going to get free points. Okay. And what if they were to say, point to the amide? You're going to circle this, right? Oh, you guys, yeah. It's just they kill the MCAT is a dumb joke, but it works. Okay. So again, more free points on the MCAT, right? Every once in a while, they'll ask these super easy questions and just grab them. Okay. So what is the significance, right, of the peptide bond, right? It's got unique characteristics, and that's because of this amide resonance. Okay. So what's going to happen here is that you get this partial single bond characteristics, right? Which makes the bond longer. You're going to get these partial double bond characteristics. right? And what's this going to do? This is going to make the peptide bond rigid and planar. Okay. And then up here, right, you have this partial negative charge, which makes this oxygen easily protonated. Okay, so these are all, peptide bond characteristics that I would be comfortable with. Yeah, so that's a good question. Dark line equals partial single bond and dotted line equals partial double bond. In reference to how it's drawn out here, right? So, traditionally this would be, can you guys see hold on, my marker keeps disappearing. Oh, there it goes. Right, so you would normally write this out as the double bond here, okay? But it has a resonance with this peptide bond here, this carbon to nitrogen, right? And so you would say that this has a partial single bond, right? Just because it's normally written as a double bond, and that this would have a partial double bond, right? Because it's normally written as a single bond. Does that make sense, Michaela? And someone asked another really good question. What's the significance of rigidity and planarity? Okay. And that's because it's going to give proteins, this defined structure, right? Where you have this backbone that doesn't rotate, right? Like chiral molecules would rotate, right? In space. So it'll give these proteins, a much more firm structure. And we're going to see that in a second when we talk about, beta sheets and actually tertiary structures and alpha helices, okay? Cool. Yeah, good questions, guys. Keep them coming. Sorry, when I said tertiary structure, I meant secondary structure. Okay, so yes, that would definitely add to it, right? So enzymes can attack the backbone on either side and cut it in different ways. But it's also important because it allows you to more easily form, right, these secondary structures, right? Oops. Okay, so when we talk about secondary structures, right, it involves hydrogen bonding, very important, between backbone atoms and not the R groups. Okay, so whenever you see a protein image, right, and you have like an alpha helix, right, which is this thing right here, okay, it's a secondary structure because you get this hydrogen bonding between the backbone molecules, which are more easily held in place because they have that planar structure. Okay. Does this all make sense? And then here, this is a beta sheet. All right. And again, here's the backbone, right? So for proteins, right, the backbone is NCC, 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 NCC. Okay. And then you have the R groups, right? These functional groups. Okay. But the actual secondary structure is referring to how these interact, the backbone. Okay. So the key here is, in case you get a question, secondary structure, not R groups, right? It's hydrogen bonding between backbone atoms. Okay, any questions? All right, let's move on to tertiary structure. Okay, so tertiary structure, right, is actually talking about the interactions between the R groups of the amino acids. Okay. So what are some of these? Okay. You could have ionic bonds. So you could imagine, right, like if you had a residue, CH2, maybe 2, right? Right, so you have a positively charged residue, and then maybe you have a negatively charged residue, right? So it's got like an O and an O, right? You'll form a ionic bond that keeps these two regions close to each other, right? Similarly, right, you could form something called, you could have hydrophobic interactions, right? Well, maybe you have a, residue, right? A non-charged, non-polar two amino acids, okay? They'll form hydrophobic interactions, right? Ugh. Okay, what's another one? Hydrogen bonding would be another one. But again, between the R groups and not the backbone, right? So you can imagine, the hydrogen bond, right? Here, or whatever, right? I'm going to specify that and say R group. Okay. Van der Waals forces too. Those would be much less prevalent. Yeah, they would be everywhere, right? That's just, kind of the catch-all for things that don't fit the ionic bond, hydrophobic interactions. Oh, well, there's one more actually that's super important here. Disulfide linkages, right? So covalent bond, right? So you can imagine your cysteine residues, right? Right, it's tertiary. Cool. Okay. What's the difference between hydrophobic interactions and van der Waals forces? Van der Waals forces, right? If we can go back, and someone correct me if I botched this, would be dealing with these partial charges right here, right? So if you have things that just carry a slightly, negative, and then maybe there's another species that carries a slightly positive, they'll end up being more attracted to each other, right? Versus hydrophobic interactions where you have non-polar hydrophobic residues being attracted to each other, right? Like these two right here. Okay. Okay, so now that we're on tertiary structure, okay, and we're talking about, when we talk about tertiary structure, we're talking about, 3D structure, right? We're based on, three-dimensional coordinates, right, of a single subunit. And so, in general, how do proteins fold? Right, we're talking about in an aqueous solution because we're in a biological environment, right? So aqueous, right? How do they fold? Yep. Hydrophobic R groups on the interior. So you can imagine if you have an aqueous solution, which means that the environment is going to have negative and positive charges, right? your hydrophobic residues are going to dislike that. And so what's going to happen is the hydrophobic residues are going to end up in the very interior of the protein versus your hydrophilic residues, which are more likely to be seen on the outside. Okay. And someone says, are the hydrophobic interactions only occurring due to protein folding from an aqueous solution? That's like a chicken or the egg. There will be hydrophobic forces, right? Especially when you get into little microenvironments of, different pockets of the protein or enzyme, whatever you're talking about. But for this situation, yes, right? You're going to want to have, just aqueous environment is going to repel hydrophobic and the opposite. So the hydrophilic is going to, want to interact with the aqueous environment. So that is what's going to cause this general folding mechanism. Okay. And so, great. Yeah, good question. Okay. And so before we go on any further, let's talk about disulfide linkages. Okay. So here's a couple of residues, right, that contain cysteine. Okay. And so what do we do when we go this way? When we form these disulfide bridges? Yeah, good. Oxidize. And so the other way, we reduce. Cool. Okay, now let me do one thing. And I don't know if this will ever come up, at least on this newer MCAT. But if you're ever reading and you see cysteine, right? So just an I versus cysteine with an IE. Cysteine is referring to the oxidized species. And cysteine is referring to the reduced species. Okay. Cool. Great. Okay, let's talk about quaternary structure. Okay, so this is talking about the 3D environment involving multiple polypeptide change, right? So they come together to form, multiple subunits, that build a functional protein or an active enzyme. Okay. And so the quaternary structure, right, refers to the number and the arrangement of protein subunits with respect to each other. Okay, so you can have, a monomer, right, which is just one. Okay, and then you can have, either homo or hetero. Right, and you can go from, a dimer, trimer, tetramer, pentamer, hexamer, heptamer, octamer, and so on and so forth. Is just going to refer to the number of subunits. Okay, so this right here is hemoglobin. Okay, right, and this is got 4 subunits, and 2 of them are homo and 2 of them are hetero, right? So these are going to be the same, and then these ones are going to be the same. Okay, so it's a tetramer. Heterotetramer is hemoglobin. Okay, and so just to give you guys a heads up, just so you can pay attention, as you do your studies and do practice questions, but like classic MCAT molecules that are in some way going to deal and be tested on the quaternary structure, right, are going to be hemoglobin, right? Right, things that keep showing up all the time, DNA polymerase, Okay, right, and maybe I'll give you a little bit of context here. Right, when we talk about when hemoglobin quaternary structure comes up, right, it's going to talk about the allosteric regulation, by things like CO2, carbon monoxide, 2,3-BPG, right? Okay, so we're not going to go over these now, but just keep in mind these are very commonly tested things on the MCAT. So you will run into them, right? DNA polymerase, right? It talks about how DNA polymerase is a holoenzyme, right? Which means when it's an active enzyme, right, with all the subunits versus when it's not a holoenzyme. Okay, another classic one, another classic MCAT structures that deal with quaternary structure is ion channels, right? These are going to be classic, right? Right, and these, the context of those is, it'll ask you, right, something like, depending on the ion channel and the different subunits, right, you're going to get, specificity for different ions. Okay. And then, and someone asked, hemoglobin, it's a heteropolymer. Tetramer, right, is how you would say it, right? So hemoglobin has different subunits, right? So there's these two that are the same and these two that are the same, but there's 4 subunits in hemoglobin. So it's a tetramer, right? Mono meaning 1, dimer meaning 2, trimer meaning 3, tetramer meaning 4, pentamer meaning 5, hexamer meaning 6, heptamer meaning 7, octamer meaning 8, and so on and so forth, right? So it's a tetramer, and because there's different subunits on it, it's a heterotetramer. Okay. Yeah, good question. Okay, and then what's another classic? Nucleosomes, right? Those are another classic quaternary structure that gets tested, right? And this is how the context is DNA packaging. Okay, right? How DNA can interact with proteins, right? Maybe it's going to talk about epigenetic modifications. Microtubules, yeah, that's another classic. Gosh. Okay. Right, and this is going to be something that, context like when it's talking about protein structure, protein as a structural component. I'm just saying these just so when you come across them, just to pay a little bit extra attention. Okay, right? And you might get questions that talk about, disrupting the quaternary structure, what that's going to do, to the context of the cell and the function that it's trying to perform. Okay. Does anybody have any questions on that? Okay. So again, these are just, classic MCAT proteins that seem to get tested a lot. And it usually is because of some disruption to the quaternary structure. Okay. Okay, let's, I think it's time for a couple questions. So let's, check our understanding here. And we'll go over these. Maybe I'll give you 2 minutes, maybe a minute and a half, right? You should kind of aim for 30 seconds per freestanding question. Cool. Maybe another 20, maybe another 30 seconds. So someone asked a good question. Are holoenzymes enzymes without their cofactors, or is that an apoenzyme? Holoenzyme, so the way to remember that, holo means whole, means with everything it needs to perform, right? So holoenzymes would have their cofactors, correct. Okay, does anybody have an answer for question 1? Cool, so we've got a few answers. We've got a couple of these. We've got a couple of these. More of these, a couple more of these. Awesome, great job. So this is a really good fundamental question. And the answer to this question is actually A, primary structure. Okay, and it's very confusing because it's very easily to think that, right, especially, we go over these slides, right? How these R groups are interacting, which is kind of holding the thing together, right? So you would think that it would be the tertiary structure, okay? But what actually it's primarily based on is the primary structure, okay? Now, this is important because this was actually a very, very famous early biochemistry experiment. And it was done by this guy named Christian. Nice, Athenson. You took the words out of my mouth. Athenson, right? And I think this was somewhere around 1961. Okay. Right. And so he demonstrated, right, that if you took an active enzyme, right? So let's say we had this enzyme right here, and it's all folded on itself. And it turned out it was actually, I think it was bovine ribonuclease, right? And what he did is he denatured it. So this was active. It works. And he denatured this protein using beta mercaptoethanol and urea, and it, denatured the protein. Okay, reduced all of these SHs, right? Obviously, this is inactive. Okay. Okay, so this is important. This is in the presence of BME denaturing, right, and urea, right? And so what he did is he took away the BME, and it reoxidized all of those disulfide bonds. Oops, let me go back one. Right, and it formed a inactive protein. But when he took back the urea and the BME, right? So now he subtracted, right? So this is minus BME. Now this is minus BME and urea. The protein actually became active again. Okay. So this proved that, you can denature a protein and then just take away the thing that denatured it, and it's going to fold back in the exact same way, right? Which suggests that protein folding based on its primary sequence is not done randomly at all. Okay. And it turns out, right, that like even if you take something like a 100 amino acid residue, okay, you end up with something like 3 to the 100 different possibilities for the final folded protein. Okay, so if proteins like literally had to randomly try to fold themselves into being active, right? You take the average time it takes for protein to fold, it would end up being something like 1.6, times, I think it's like times 10 to the 30 years, right? And it turns out that the universe is actually not even that old, right? Only 1.5 times 10 to the years old, okay? So it's the primary structure that determines the protein folding. And then those, R groups that'll actually stabilize it that way. Does that make sense to everybody? Oh, and Tiffany, I have these questions, right? Do you want, I can send them to you after. Do you want them? No, the R groups do not determine folding, right? Does that make sense? Yeah, it's the primary structure, right? So the idea is that if you had like a native active protein, right, and you were to randomly form disulfide bonds, it would be inactive, right? But if you were to let it just fold on its own, it'll, whether you can, reduce it or oxidize it, right? It's reversible. And so the protein would become active, which means that it's the primary structure that determines the folding, not those R groups. These ones, there might be one of these. These are like pretty, these questions might be in the book. A lot of these questions aren't in the book. These based on non-descriptive passages may or may not be in the book, right? But I can send them to you if you want. Okay. All right, what about question 2? Yeah, I can send you guys the questions after. As long as this question 1 makes sense, right? Fundamental experiment by this guy. So we got a couple for A, and then we got a couple for B. Okay, nothing for the other ones. Good. So the answer is actually B, hydrolysis of peptide bonds. Okay. And so right now, obviously the class split between A and B, right? It's just like, take your best guess, right? What's the difference between hydration and hydrolysis? Hydration, right? This is when, you combine something with water. But no bond splitting. And then hydrolysis, right? It's where water breaks the bond. Cool. Right, so like the general reaction, you can imagine is like R1, OR2, right, plus H2O, 2, R1, OH, plus ROH, sorry, R2OH. Cool. Okay, and someone asked another good question. Can you explain what you mean by protein catabolism? Okay, we're going to cover this when we talk about metabolic pathways. But just to give you, clue in, when you talk about catabolism, you're talking about breaking down something, right? So in this case, protein catabolism is breaking down a protein. So starting to chop up all those peptide bonds and using them and recycling them in some other way. Okay. And so if we're talking about protein anabolism, it would be actually building the protein, right? So, you'd actually have your tRNAs, actually adding on residues to a chain. Okay. Cool. Yeah, exactly. Exactly. Yes, and anabolism is analogous to metabolism in respect of, so anabolism is building up, catabolism is breaking down, right? And we're going to go into we're going to talk about that a whole lot. I mean, we got next lecture, talk about glycolysis. A couple of weeks after that, we're going to dive into, metabolism. So don't worry about that too much. Okay, cool. Let's talk about carbohydrates and lipids. I don't want to jump into this stuff too much. Just maybe do we're going to just, do one dense slide for each. And that's just because we're going to talk more about them, as they come up in examples as we go along. Okay. But, at least for now, we're talking about, structure and function and something to build off of. Right. When we think of sugars, for example, right, we want to ask, what's it being used for in a biological context? Okay. Right. So is the sugar, used for energy metabolism? Is it used for energy storage? Is it used for, structural support? So those are things that you just, keep in mind as you, hear the word sugar, right? Generally, this is the empirical formula for sugar. Like I said earlier, there's the simplest one that follows these rules of needing to have at least an aldose or a ketose and, one or more alcohol groups, right? It's a 3-carbon sugar. And they go up to 7 carbon sugars in your body. I can't remember an example of one, but most of the time we're talking about 3 carbon, 4, 5, and 6 carbon sugars, right? And each one will play a slightly different role within your metabolism, which we'll talk about later. Okay. Monosaccharides, right? These are just simple sugars, so they're just one ring structure. Disaccharides, it's anything that has, 2 ring structures. So like lactose would be a good example of a disaccharide. It's got a glucose and a fructose attached. Monosaccharide, glucose, blah, blah, blah. Oligosaccharide. These are, again, just like oligopeptide, right? Oligosaccharide is a short chain of sugars, right? And specifically, right, if you ever hear oligosaccharide, I want a little light bulb to go off and say, okay, are they talking about glycoproteins? Or are they talking about glycolipids, right? A glycoprotein is just a protein that has sugar, an oligopeptide attached to it, right? So, a short chain of sugars. And similarly, a glycolipid is a lipid that has a short chain of sugars attached to it. Okay. And, later you'll realize that these sugars will play actually an important role, and they might alter the function of the protein, right? They might alter the solubility. They might, change the binding affinity of different proteins for enzymes just by attaching or detaching these oligosaccharides, right? Okay. And then polysaccharides, these are just, long chains used for, most often energy storage, or if we're talking about plants, sugar, right? So like starch, glycogen are energy storages, and then like chitin and cellulose in plants, which cellulose is actually the most abundant organic molecule on Earth when we're talking about polysaccharides. Okay. And again, we're going to talk about those, more just as they come up. But lipids, right? Again, super basic overview, right? Lipids can dissolve in non-polar solvents, but they're insoluble in water. Okay. And so, for biochemistry specifically, we keep running into, these five, basic structural building blocks of lipids. Okay. And so, as we proceed, you're going to see the importance of each one of these. So just, again, keep them in your mind, right? I'm sure you guys are aware, fatty acids, carboxylic acids with long 16, 18, typically, carbon chains. They can be saturated, unsaturated, right? You guys should kind of, know all this. All right. And so, we'll get more into the specifics and the functions of them as we go. But this is just, again, now they're in your head, now what do each one do? What's the significance of each one as they come up? Okay. So we'll move on now to nucleic acids. All right. And so let's talk about, the structure of the nucleic acid, and let's talk about the nomenclature. And believe it or not, for some reason, MCAT test writers Oh, yeah. I could do that. So what I do is for my slides, if people request them, I'll send them, but I'll dumb them down, right? I don't like giving people the entire slides, one, just because then they're out there and people tend to show up to lecture a little bit less. So I like to force that issue. And two, you'll take better notes. So I can give you like a slide outline. I could do that. Yes, hold on. Okay, I just sent something over to you guys. Let me know if it comes up. I could do it as class starts, or I'll just put them up on the, actually, I'll do it as class starts. I don't want to put them on the portal just because that way you'll get the class on time. And I'll make a deal with you guys. If you get the class on time, I can upload the slide outline. Okay. But let me know if this gets through to you. And I understand this is a lot of information, right? And so the slides are actually nice. And it's nice to take notes. I don't know if you guys take notes on your iPad or if you take notes on cool. Do you guys get this one? Yeah, people take notes on the iPad. It's nice to have the slides so you can actually just write over them. And to be honest, we go through so much material. It's so hard to write all this down. Okay. Yeah, I'm actually a paper person myself. I love using a pencil. I'm taking a little trolley break right now. Sorry. Did you guys get that, by the way? Okay, great. We can move on. So with nucleic acids, right? These MCAT test writers, they absolutely just love to test the structure of them, right? And you get like one question, probably, maybe even one or two, probably just one, but it seems like every single MCAT ever has had some sort of question on nucleic acid structure. right? So it's, you want to collect that free point. Okay, so with nucleic acids, okay, you have, the phosphate groups. Okay, you've got the pentose sugar. And then you've got the nitrogenous base. Okay. And so these are the characteristics of a nucleotide. And if you wanted to take this further, you could even label the carbons of the sugar, right? Okay. And so what they'll test you on is the nuances between different nucleotides and nucleosides. Okay. So for example, probably the biggest one that they do is what's the difference between a nucleoside and a nucleotide, right? A nucleoside is anything that doesn't have one of those phosphate functional groups attached. A nucleotide is anything that has one or more phosphate groups attached, right? So you can imagine, this is a nucleotide, this is a nucleotide, and this is a nucleotide. Okay. So far, so good. Right. And these again are just free points. And now if you look at the ribose ring, right? Deoxyribose, right? Which is, the one used in DNA, is going to have a missing hydroxyl group at the 2' carbon, as opposed to the ribose ring, aka RNA, which has both of them intact. Okay, and then later we're actually going to talk about dideoxyribose, which is going to have both of those gone. Okay. Other things that you need to know are going to be the bases, right? So these are your nucleotide bases, A, G, C, T, and if we're talking about RNA, U. All right. So, it's a good idea to know what they are. And I think there was the clever mnemonic, right? I think it's in the handout. Does anybody have one for these? How do you guys remember which one is a purine and which one is a pyrimidine? Yep. All gold is pure, right? Pure is gold, right? So purines, all gold is pure, right? So and this one's a little sillier, right? It's time, it's time to climb the pyramid. Right? Cut the pipe, cut the pie. Oh, cool. Yeah. Anyway, it's good to know these, right? Having these little mnemonics is fun for when you have a brain freeze, and then you just have to, flip through your brain files and remember what they are. Okay. Now, right, purines and pyrimidines, they form bases through hydrogen bonding. Okay. So A and T, right, can form 2 hydrogen bonds. Right? Same with A and U if we're talking about RNA. Right? And then guanine and cytosine can form 3. So GC. All right. Okay. So, right, DNA polymers, right, they're built from deoxynucleoside triphosphates, DNTPs, right, which is what we just went over. Okay. So if you take here, right, let's say this is a replicating DNA strand, right? It's the 3' hydroxyl that actually is going to attack the alpha phosphate of the new base that you're going to add, and you'll break off these 2 phosphate groups, right? And then you'll add, 1 base pair to your growing sequence. Okay. So this 3' hydroxyl it's key here for DNA replication. All right. This should all be reviewed. So let's move on to a couple more questions. All right. Maybe we'll take a minute and a half for these, and then throw your answers up. Maybe 30 more seconds. Any more guesses? So we're a little bit all over the place so far. Anybody else? And then we have, for 4, we have a B. Oops. And then a bunch of no ideas. Got a 3D, 4B. Okay, cool. This is great. So let's go over the first question, right? And this is actually, another, I keep using this a lot today, but, free points on the MCAT. Okay. And yes, it is one of those wretched accept questions. All right. Which means that, have you guys heard of Chargaff's rules when we're talking about DNA? So this is something that we should go over, right? So when we talk about DNA, there's something called Chargaff's rules. And it's this proposal, right, that your percent A nucleotides is going to equal your percent T nucleotides, and that your percent G nucleotides are going to equal your percent C nucleotides. And you guys should write this down in your notes. And that A plus T is equal to T plus C. Okay. And that is because for every A, it's going to hydrogen bond to a T on the other strand of DNA. And for every G, it's going to hydrogen bond to a C on the other strand of DNA. Okay. And therefore, total A plus G is equal to total T plus C. Okay. No, A plus G is equal to T plus C, right? So if you look at, oh, wait, no, you're right. Did I write T twice? Oops. A plus G is equal to T plus C. Sorry. Okay, and that's because this plus this, A plus G, right, is going to equal to T plus C, right? Because this binds to this hydrogen bonds to this. Got it? And so if we go through question number 3 now, right, this is a good question because it asks you, right, which ones are the purines, right? And, you know, you have to know A and G, right? Which ones are the pyrimidines, right? And you have to combine them. Okay. So which ones are purines, right? A and G. So total purines equals A plus G. That checks out. Okay, so this is correct. And then because it's an accept question, you can just cross it right off and be done with it. Okay. And so does total purines equal total pyrimidines, right? So that's A plus G is equal to C plus T, right? Check, check. So this is correct. So now we can eliminate choice B. And then A equals T and G equals C. A equals T and G equals C. That is correct. And so the last one doesn't make any sense. Does everyone see that? Great, thank you. Let's talk about question 4. Now, this is another good question. And this is a good question because in classic MCAT style, it's giving you a bunch of extra information that you absolutely don't need to know about. Okay? Right? It's talking about E. coli, and it's, you know, E. coli's got this, you know, fancy circular double-stranded DNA chromosome, right? And it contains, you know, all these base pairs, right? And then it's giving you the specific count for, you know, that there's this higher concentration of the GATC base pairs, you know, within that chromosome, right? And then it asks the fundamental part of the question, right? You know, whereas the expected GATC frequency in the whole chromosome is 1 in, and then it gives you all the answers, right? So what is the expected frequency of G, right, to pop up, right? There's only 4 nucleotides, so there's 1 in 4. What about for C, right? 1 in 4. What about for T? 1 in 4. What about for A? 1 in 4. Which means, oops, which means if you multiply these together, right, because we're talking about probability, it's going to be 1 in 256 base pairs. Cool. Do you guys see that? Do you see that the answer is C? Great. Now, I mean, while we're on the topic of DNA sequences, right? And this might be getting a little bit ahead of ourselves here, but when we're talking about PCR and all these, you know, molecular biology lab techniques, you know, that require primers to do their, you know, DNA replication or PCR, whatever it is, right? That's why they design primers to be somewhere between, you know, 10 to 20 base pairs long. And that's because if you were to take the probability of running into that exact same sequence, it gets astronomically high really quickly. Okay. And someone says, we do 1 in 4 because we're trying to get a frequency of 1. We do 1 in 4 because there's only a 1 in 4 chance of each base pair, right? So if you have A, T, C, G, those are the 4 nucleotides, right? You have a 1 in 4 chance of running into this one, 1 in 4 chance of running into this one, 1 in 4 chance running into this one, 1 in 4 chance running into this one. All right. All right, I'm gonna move on just for the interest of time. Okay, nucleic acids, DNA versus RNA. Okay, you guys probably obviously know RNA has uracil in place of thymine, right? Okay, but why is that? Why does RNA have a different base pair than DNA? Okay, this is actually a question that's not completely answered within the scientific community, but one of the hypotheses is that cytosine can undergo a deamination, right, which will actually turn it into a uracil. Okay, so you can take this C and it can accidentally get deaminated, and it'll turn it into a U. So you can imagine if DNA is so important to survival, right, that those DNA repair mechanisms had life not evolved to have this new genetic code from RNA, it would have a much harder time repairing DNA. Okay, and so if we were to compare RNA and DNA from a structural standpoint, right, RNA compared to DNA, RNA is much more unstable compared to DNA is more unstable. Right? It doesn't form this double helix, right? All it can do is kind of fold back on itself, which you can see here through this hairpin, right? Which is very minimally increases the stability, right? RNA, if it does fold back on itself, has larger grooves, which means that if an enzyme comes to bind and cut it, it can do so more easily, right? So RNA is just a much less stable molecule than DNA. Okay. Okay, so the double-stranded DNA now, when we're talking about its stability, right, there's a lot of different factors that will affect it. Okay, so if you ever hear the word pairing, it just means the same thing as hybridizing, right? So DNA can pair with another strand, DNA hybridizes with another strand, or DNA anneals with another strand. Those all mean the same thing. The two single-stranded DNAs come together and they form a double-stranded DNA. Okay. If you see this fancy word TM, right, temperature melting point, right, this is the temperature at which 50% of the DNA is denatured. Okay, and as you can see from this graph here, right, the higher the temperature, the more DNA will be denatured, right? So you'll go from double-stranded to single-stranded DNA. All right. GC pairs, those increase stability. Why? Because GC pairs can form 3 hydrogen bonds. So if you have more GC pairs in a genome, then you'll have to warm your DNA up to a higher temperature for it to denature. Okay. Salt concentration, that will increase the stability of your DNA. Okay. And that's because you can imagine if you have DNA, right, and they're negatively charged. If you have a positively charged ion like magnesium, it can actually help stabilize those negative charges. So the negative charges from DNA next to each other have a less of a repulsion of each other, right? So the backbone is more likely to stay together. I mean, the DNA is more likely to stay hybridized. Okay. This should all be pretty good review. So we'll quickly move on to cellular membranes. All right. So what is the main component of cellular membranes? They're phospholipids, okay, which are amphiphilic molecules, right? And what does that mean? That means that one part of the molecule has an affinity for polar solvents, and the other part of the molecule has a affinity for nonpolar solvents, right? So in other words, when you make your, lipid membrane, right, you have the lipids, nonpolar, polar. Okay. Which is what leads to cell structure, right? Cell membrane structure. It'll lead to these things called liposomes and micelles. Okay. And so what happens if you were to put these phospholipids in an aqueous solution, all of a sudden they're spontaneously going to aggregate, right, to sequester these non-polar tails. Okay. So they're either going to form a micelle or a liposome. And in the case of a cell, it forms this bilayer sheet. Okay. Why are liposomes so important? What's so cool about liposomes? These are super important for the context of our MCAT, specifically because the liposome, exactly. Thank you so much. Drug delivery, right? So researchers have increasingly adapted liposomes as a way to deliver drugs within the cell, right? So you have a drug, it's really hard, it can't get past, the cell membrane. What you can do is you can package a drug into a liposome and get it to actually go through or I forget the word, but bind to the membrane and actually get that charged molecule into the cell for drug delivery. Exactly. And my cells, it could do that with things that are hydrophobic, right? So you could throw a hydrophobic molecule in there. I think an example of that would be like vitamin D. Is hydrophobic, so you could put that into a mouth and actually get it to go in. Okay. All right. So membrane fluidity. All right, there's lots of different factors that will actually affect membrane fluidity. And you've heard this a million times, but it's good to go over it again just so we can move on from it. Right, just because it's assumed for everything that comes after this. All right. So what do double bonds do? Right? Double bonds on your phospholipids. Those are going to increase fluidity. And if anyone wants me to explain why, I will. Otherwise, we'll just move quickly through this. Cis double bonds specifically will increase fluidity even more than trans bonds. Temperature will increase fluidity. Yes, absolutely. So if you could imagine a cell membrane, right? Maybe I have a drawing of a cell membrane here. So if you have a cell membrane and you have these phospholipids where you have like the phosphate group, it's negatively charged, and it's attached to these fatty acids. This is a crude drawing, right? So these are carbon chains. This right here is going to be a saturated, right? So there's no double bonds in there. It's just saturated with hydrogens, right? You could actually stack these much closer to each other than, say, if you had a phospholipid right, where all of a sudden you have a double bond, right? And it sends your tail off this way, right? So now if you have these phospholipids with tails, right, they're going to take up a bigger surface area and they're going to run into each other and they don't stack quite as nicely. And so it'll increase the fluidity. There'll be more available area for them to move, right? Does that, Melody, does that explain it for you? Great. Yeah, very important. Okay, now this is where things get weird, right? What about cholesterol? Okay, so here's cholesterol down here, right? What does this do to fluidity? We've got one for decreases fluidity. Anybody else want to give it a shot? You're not wrong. You're not wrong about decreases, but you're also not right. Cholesterol, interestingly enough, is actually a bidirectional regulator of membrane fluidity. Okay. So cholesterol, maybe I should write this down. Let's give me some room here. Right. Cholesterol at high temps does what? It decreases fluidity. And cholesterol at low temps increases fluidity. Okay. Now, how that works is cholesterol is this very planar molecule, right? And so it can actually fit in between, as you can see right here, these phospholipids, right? And cholesterol, this picture is very inaccurate. Cholesterol actually makes up something like 30% of most cell membranes. Okay, and so it'll actually disrupt those interactions between these phospholipids. Okay, and it'll create this phenomenon where it's essentially acting as a buffer, right? So it'll increase or decrease the fluidity as it's needed. Okay, and so I guess the thing to know and why you guys say decreases, and that's because at body temperature, so at 37 degrees Celsius, cholesterol will actually decrease fluidity. Okay. So is everyone satisfied? At body temperature, cholesterol will decrease fluidity. However, it acts as a bidirectional buffer. So at a higher temperature, it'll decrease the fluidity. If the cell goes onto a lower temperature, it'll increase the fluidity. Tiffany. Okay. And so why is this so important, right? Why do we care about fluidity, right? And that's because you need your membrane to be fluid enough so that new proteins, right, membrane proteins can be inserted into the bilayer. Okay. It also needs to be fluid enough that, if you have two different, maybe you have like a liposome coming to fuse with the membrane, that it can actually do that. Okay. And it also is very important because it ensures that molecules can be distributed evenly, right? They can move around and kind of float around, and then things, by entropy will randomly just, spread out evenly. So you don't have these clusters of certain things on any given side of the cell. And someone says, what about trans double bonds? Okay, so good question. You guys have all this? I'll just go back one. All right, so if you're looking at a double bond, right? That's a cis double bond, and that's a trans double bond, right? So these cis double bonds in the context of a hydrocarbon chain, oops. No, I don't want to draw a straight line. Will actually bend the molecule more, right? So it'll have a bigger angle here. In cis, trans will have a smaller angle. Does that make sense, Brett? Awesome, great question. Okay, let's keep going. Let's talk about diffusion, right? Another very fundamental, basic, probably review concept, but it's just good to know and hammer in because everything will build off of it, right? So we're talking about, cell membranes, right? The rules are that small non-polar solutes right? So like oxygen, carbon dioxide, nitrogen, right? Non-polar steroid hormones, these can diffuse directly across a plasma membrane. Okay. And then things that are large and or polar, like ions, water, glucose, right? Things of that nature cannot. All right. I mean, just like take a mental picture of this. Okay. Okay. And, membrane proteins, right? There's different classes of them. There's integral, right? So these are going to span the bilayer. Right, so which ones are integral? This would be an integral, right? This would be an integral. This would be an integral membrane protein. There's peripheral. Right, so loosely attached to the outside. So maybe we'll make this like green. Right, so this will be a peripheral, peripheral, peripheral. Okay. Peripheral membrane proteins. And then there's glycolipids and glycoproteins. And we kind of talked about this earlier. Right, so this is going to be any of these proteins, or maybe it's a lipid where you attach, in this case, it's, sugar molecules, right? These branched sugar molecules to the lipid or the protein, and that sticks into the lipid bilayer. And you don't need to know this yet, but, this will be important for, cell signaling and communication and binding sites that are sticking out of the cell membrane of the cell. Okay. Anybody have any questions on that? This is just, some naming, just so if it comes up in a passage, you know what they're talking about. Let's talk about the different types of membrane transport. Okay, so the very basic one would be simple diffusion. Okay, and this is what happens when you take, these small hydrophobic, molecules that can diffuse really quickly. And simple diffusion happens when you go from a high concentration to a low concentration. So you go down your concentration gradient. Okay. And so the ones that will do simple diffusion the fastest are those small hydrophobic molecules, right? And then larger polar molecules will diffuse, very slowly. So for example, sugar, it'll take, a very long time for sugar to diffuse down its gradient. And then things that'll take even longer, so very, 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 very slowly, will be, your ions, right? Something with a charge, your sodium, potassium, calcium. Okay. So that's simple diffusion. Oh, and someone asked a question. Are integral membrane proteins also known as transmembrane proteins? Yes, they are. Okay. Right. So integral, yes. Integral proteins will span the bilayer, right? And then transmembrane proteins, they're on both sides. Okay. Right, so integral And then these green ones are peripheral because they're just very loosely attached. Okay. Good question. Thank you. Okay. So we went over simple diffusion. Okay. And so simple diffusion is going to be a type of passive transport. Okay. Which means it does not require energy. And so another category of passive transport is going to be something called facilitated diffusion, right? And facilitated diffusion is you're going to need these transmembrane proteins, right? And the interactions between transmembrane proteins and specific, molecules is going to dictate its ability to go through. And again, for facilitated diffusion, okay, it goes from a high concentration to a low concentration. So with the concentration gradient. Okay. And there's three, types of diffusions, right? You can have a uniport, a symport, and an antiport, which just as the name implies, an antiport, you'll take a molecule and it'll move it out for every molecule that goes in, right? A symport, you'll couple a molecule with another molecule and they'll both go in. And then a uniport, you just have one molecule that goes in and passes through. Okay. And so these things can be very specific. In fact, some things are less specific, but some of these, transmembrane proteins can be highly specific and only let a certain type of thing go through. Okay. So contrary to passive transport, you get something called active transport. Okay. And this is when you're moving, molecules, ions, whatever it is. Oh, I just got a message from someone. 1 second, sorry. One second. Okay, thank you guys. Okay, and then, so contrary to passive transport, sorry about that, is something called active transport. And this is things that go against the concentration gradient. Okay, so these are molecules that require energy to get transported in or out of a membrane. Okay. And again, these can be, very specific. And so there's two categories of these. There's primary active transports, and then there's secondary active transports, right? And the classic example of a primary active transport would be, hydrolyzing ATP, right? ATPase, and the energy gained from that will actually transport, it'll move 3 sodium molecules out of the cell. At the same time, it'll move 3 potassium molecules into the cell. Okay, and it'll burn an ATP doing that. And so that would be an example of primary active transport. Now, secondary active transport, right? So in this exact same example, notice how we're pumping sodium outside of the cell, which is actually going to give the cell a net negative charge, because for every 2 positive charges you pump in from potassium, you're pumping out 3 positive charges, okay? Which means that you're going to create a gradient between sodium on the outside of the cell over here versus on the in. And so what secondary transport is you can couple things like this, in this example, the sodium gradient, right, which wants to travel back into the cell with its gradient, and you can actually couple that and move, molecules like glucose in. Okay. And this is considered active transport because the cell is going to have to keep this membrane potential and pump out that sodium, which is going to take ATP. Okay, let's pause there for a sec. Does that make sense? Yep. And someone says that's exactly right. So the very classic example, this very fundamental, it's called the sodium pump, right? Or the Na plus K plus ATPase pump, right? Which actually burns something like 30% of your energy, I just read, in your brain and in other items, just to create this membrane potential. Moves 3 sodiums out for every 2 potassiums that move in. Okay. And directly burns ATP, making it primary active transport. Yeah. And so secondary active transport, right, takes advantage of the fact that there's excess sodium on the outside. So if this is outside, all of a sudden we're creating a gradient. So there's excess sodium. And so sodium will want to go back into the cell, right? And so that energy can be saved and coupled with transporting something that would be harder to go into the cell by itself, like glucose, and move both of them into the cell. Okay. Right. Which you would think, oh, that doesn't take any energy. The problem is now you have your sodium back down here, which needs to be pumped back out of the cell. So you do burn ATP doing it. Okay. Very good question. Very fundamental. Does anybody else have any questions on that? And, this is going to become very important in some of your physiology lectures when we're talking about, synapses and action potentials and stuff like that, right? And you're going to actually visit, and you should know these numbers. You don't need to know them just yet. The resting cell membrane, cell potential, right? The concentrations of all of these ions in and out of the cell. Okay. So let's do another question. I'll give you guys a few minutes for this, maybe 3. I know, obviously, this lecture is going to go just way over time. So if you have to go, that's totally fine. We can make it up later, come to office hours and go everything. Again, everything is pretty basic, especially after this. We're just going to brush over, things that you can read out of the book. So it's not super critical. But if you do want to stick around, I will keep going. I don't know. I would say probably, 30 minutes, maybe, honestly, maybe even an hour. The second half of the lecture moves quicker. Yeah, it's just much, right? This is all the, it's a heavy review, but all these things, right, are what is assumed that you guys know for everything else that you have to learn. Okay. And yeah, while you guys read this problem here, I'll just type it actually. Take another minute on this question and then let me know when you guys get some answers. Does anybody have any answers? Does anybody need more time? Cool. Let's start going over this. Just so I've gotten a bunch for 5B, 5D, something like that. 6D, D, right? Great. Yeah. So the answer to question 5 is actually D, alanine uptake. Okay. And it pretty much says all of this, in the passage, right? So this is, on the MCAT, you're going to need to be able to read through these things and read through them pretty quickly, right? And kind of understand what it's saying. And, these answers you could pretty much take directly from the passage. But again, you need to know, right? The more you know about the certain structures and mechanisms and enzymes going into the passage, the quicker you're going to actually be able to decipher it. Okay. So, right, what is this question asking? Would you expect the following to increase in mice fed ovabane? I don't even know how to pronounce this. Ovabane, right? Except, yeah, right. Again, looking for that one out. Okay, so what is going on here, right? So ovabane is this steroid that inhibits this enzyme that we were just talking about, NAK ATPase. Okay, so if you have this thing and it's inhibiting that enzyme, what that means is, if we go back one to here, right, it means that sodium is not being able to leave the cell. So we are therefore not creating that sodium gradient across the cell membrane. You're not creating that negatively charged membrane potential, right? Okay, so that is the big takeaway here. Okay, and so what does that do for everything else? You would expect all the fun to increase, right? So muscle contraction. And maybe we're jumping the gun, but as you'll learn in your physiology lecture, actually, I believe this weekend, you're going to find out that calcium, binds to troponin in muscles, and it'll cause a muscle contraction. Okay. And so somewhere in the passage here, I'm going to have to skim through it. Down here. Right. So when you get this, intracellular Na to increase, right, it'll turn on this sodium to calcium exchanger. Okay. And the sodium to calcium exchanger, as the name implies, is going to exchange, right, a sodium molecule for every calcium molecule. Does that make sense? Does it make sense what an exchanger would be? Okay. So if sodium is getting stuck in the cell doesn't like that, and it's going to need to find a way to get it out. So it'll turn on this sodium calcium exchanger, right, as a way to get the sodium out of the cell, right? So that'll increase the calcium concentration in the cell. And so the calcium, right, will lead to more muscle contractions. Okay, so this checks out. Blood pressure. How does this affect blood pressure? Does anybody know? Yep, it will increase blood pressure, but how? Need a place to draw this, kinda. But if you could imagine, here is a vein, and you've got epithelial cells lining your vein or artery or whatever it is, blood vessel, right? and all these cells are now retaining sodium, right? Then all that water that's in the blood vessel is actually going to be retained into these cells, increasing their size, which will then narrow the blood vessel. And what that does is it'll increase your blood pressure, right? So blood pressure checks out. And as we just said, intracellular calcium, right, by the exchanger is going to increase. So all of these we do expect to increase, which would make D the best answer. Does anybody have any questions about that? Cool. Okay, what about question 6? This one should be a little more straightforward. Maybe I should erase this stuff. It's an antiport for sure. Great, so we can get rid of symport. Is it directly burning ATP? No. Is it passive? Right? Let's go back to here. It's not, because we're going to eventually need to get these calcium out of here. So it's actually not passive, which makes D the best answer. And this might not be the fairest question. They might have to give you a little bit more information. But D is the best answer in this case. Yes. So someone says in the passage, it mentions that the transport is driven by the sodium gradient, right? So exactly. So it creates a sodium gradient. So it couples the sodium, right? Na here, oops, with moving, through a thing with calcium coming in. Right, which would mean that it's going against its gradient, which means that it's going to have to do the reverse, aka pump it out. So that's what makes it a secondary versus a passive antiport. Oh, and then Noah asked another really good question. Do we need to worry about why D is the answer? Or just that all the other ones, you can rule it out for question number 5. And so both is right? Alanine uptake actually is directly in the passage somewhere. Let's find it. Drives amino acids. Keyword here, right? So the amino acids are driven in the same way, if we go up to here, right, that this sodium glucose is driven, right? It can take advantage of this gradient and drive amino acids in, right, and other things. Cool, right? So that's why alanine uptake, right? So you would actually expect it to go down if you're having this inhibitor, okay? And this is why the sodium pump, as it's called, uses so much energy. It's just such a fundamental thing because it's creating this gradient where sodium's getting pumped out of the cell. So the inside of the cell has actually got this net negative charge, right? So things can actually get transported back in, okay? Which by secondary makes you burn a lot of ATP. Okay. Anybody have any questions on that? Great. Let's move on to eukaryotic cells. Now, as a caveat for these next, couple sections, I mean, we're going to just totally gloss over eukaryotic cells, prokaryotic cells, and viruses, right? Just to give you like a little foundation, and just know that you're going to be learning so much more about each of these, as you do the course and as you do, questions specifically. But these are just the things that you absolutely need to notice so you can start building off. Okay. Eukaryotic cells, right? We all know this. They have membrane-bound organelles. Okay. And it turns out that this is going to be incredibly important when we talk about cell biology, molecular biology, how each of these different organelles are kind of isolated within the cell, within the cytosol, and they serve all these different functions. And we're going to learn that, different, effectors, different hormones, different molecules will disrupt the function of these organelles or aspects of the organelles, which is going to cause serious cellular problems. It's going to cause diseases. It's going to cause cancer. Okay. And so we're not in this lecture going to go through every single one of these organelles, but there is a handout on the portal under this lecture that's going to give you a paragraph on each organelle. And I really do highly recommend reading it. And your goal should be to have a 1-sentence summary of each one. Okay. And that would be a really good, high quality understanding of, what the organelles do. Okay. So, when you get the time, do that. Right. And then you're going to find out later, just by going through passages, why all that's important. Okay. So just to give a quick, reproduction overview of eukaryotic cells, right? Human gametes, sperm or egg, they have 22 autosomes and 1 sex chromosome for a total of 23 chromosomes, which would be considered haploid or N. Okay. And so human somatic cells are diploid in that they have 23 pairs of chromosomes or 2N. So they have a set of chromosomes from mom and a set of chromosomes from dad. Okay. And so once that zygote is formed, right, cells will have 2 copies of each chromosome, right? And that's when you're now considered diploid. And so humans have a total of 46 chromosomes. Okay. And DNA non-dividing cells, right? So in the interphase section of the cell cycle, DNA is more or less linear. It's double-stranded. It's unpaired and it's wrapped around these things called histones, which we'll talk about. All right. And then in the replication phase, the S phase, right, of interphase, your DNA is going to double, right? So you'll have a total of 92 sister chromatids, right? Which is going to still equal just 42 chromosomes, right? You'll have, a copy of them, but you still only have 2N, right? You have 1 set from mom, 1 set from dad. Okay. And as cells prepare for division, right? In the growth 2 phase, this is when your chromosomes start to become really condensed and tight, and they, form that traditional, X structure that you've seen probably a billion times. And that's when they're actually, condensed and called what we call traditionally a chromosome. Okay. And it's mitosis, that is another word for nuclear division. Okay. So cells will go from 2 copies, they'll double to 4 copies of all the DNA, and then they'll split it into, 2 cells each with 2 copies. And then meiosis, this is how you form your eggs and your sperm, right? And there's 2 phases, and you go from, 1 2 copy to 1 4 copy to 2 2 copies to 4 1 copies. Okay. And, this is all just the super high level overview. And, this is one of those things that it's just better to just read in the book, right? There's all these little details about, stages of mitosis, stages of meiosis, and you do need, to know them, right? And generally I would say the important thing is to know, how many copies of DNA are in each stage and like the main point of what's going on. Okay. So, the cell cycle, which we're going to again come back to and come back to and come back to again and again and again. You can break it down into these four main phases, right? Your growth phase, your S phase, which is where your DNA actually replicates, your growth 2 phase, which is after your DNA is replicated, and then your mitosis, where your cell divides. Okay. And it's probably a good idea to have a general understanding of how long these take. 8 to 10 hours. Replication usually takes somewhere about 6 to 8 hours. This is for your average, human cell. Growth 2, I think that's 2 to 6 hours. The mitosis is the smallest one, takes about 1 hour. Okay. And just for high level understanding, before the cell will proceed into the next stage of the cell cycle, right? It's going to pass all these different checkpoints. Okay. And we're going to learn about that, what happens when a checkpoint fails, right? The cell starts to, either not replicate or replicate too much. And then you lead to things like cancers and stuff like that. Okay. And so for now, just know the general phases of the cell cycle. For now, know that interphase is G2, G1 to the end of G2, right? And then mitosis, cell division is this. Okay. Got it? Okay. And so again, super high level. We're not going to get into the weeds of this. I do recommend reading. The book goes through a lot of details in the structure and function chapter of the book. And then, when we do reproduction and development, there's going to be, another chapter on that. Okay. But generally speaking, right, if we're in, interphase, right, you're going to have your replication. So you're going to go from 2 copies to 4 copies. Prophase, you're going to have 4 copies. Metaphase, you're going to still have 4 copies. Anaphase, the cells are going to, start to pull apart, but you still have 4 copies. And then telophase, you're going to go from 4 copies to 2 copies. And then cytokinesis, you're going to have 2 copies, and now you have 2 new cells. Okay. For meiosis, similar story, but done twice. Okay. You start with 2 copies, and you're going to go to 4 copies, right? Now you have 4 copies over meiosis 1, and you're going to split that up. So now you have 2 cells, both with 2 copies of the DNA. And here's, a critical difference between meiosis and regular cell growth is that there's no S phase, which is the replication phase between M1 and M2. Okay. And so from here, you have 2 copies. You have 2 cells with 2 copies. And then it's going to divide, and you end up with 4 cells with 1 copy. Does that make sense? Oh, and someone wants me to go back. Okay. And this is just super high level, right? You need to know more than this, but it's really not, worth your time. You just going through terms and what each one does. So I do recommend reading the chapter in the book and getting, a little bit more details and just having notes, right? But this is the gist. You just need to know we're making DNA, we're dividing, right? DNA copy number and DNA like chromosome number, right? Does it have mom and dad? Does it have both? Does it have half, right? Okay, so prokaryotic cells. The big takeaway of these is that they have no membrane-bound organelles. So everything has to happen in the cytoplasm. Things actually do tend to get stuck in different areas of the cell, but there are no membrane-bound organelles. Right, but prokaryotes still have a plasma membrane, and they have cell walls that consist of peptoglycan. We'll talk about why that's important in a sec. Prokaryotes have circular double-stranded DNA chromosome, right? So it's literally a circular DNA, right? Here's your double-stranded DNA. Okay, so it's not the traditional linear, X chromosomes that eukaryotes have. Okay. They also have plasmids, which are much, much, much smaller little circular pieces of DNA. Not all of them do, but a lot of them do. Okay. And these contain, genes that might have antibiotic resistance or some sort of other advantage on them, maybe some sort of immunity where the plasmid carries something that can, depending on the environment that the prokaryotic cells, grown in, can give it some sort of, rudimentary bacteria immunity, right? Depending on the genes that are on those plasmids. Okay. Right. The DNA replication starts at a site called OREC. And this is included here just because different types of bacteria will have these different names, meaning, differentiating what they need for themselves to grow, right? Are they an obligate aerobe, meaning do they need oxygen? Are they an obligate anaerobe, meaning they can't survive without oxygen? Are they a facultative anaerobe, right, where they can use oxygen but can't survive without it? Right, so you'll see bacteria named with all these, things describing how their metabolism works. Okay, and for the MCAT's sake, and for really molecular biology's sake, when we talk about bacteria, they're so useful because you can create these species that are auxotrophs, right? Which means that it's missing something that's critical for it to survive, which is the setup, for a lot of, genetic engineering experiments. It's the setup for, testing metabolic pathways. And we'll get into that. But first, let's talk about prokaryotic cell walls. Okay. And so there's two main classes of prokaryotes. There's gram-negative and there's gram-positive. Okay. Gram-positive prokaryotes have this a much thicker peptoglycan layer that's on the outside of the cellular membrane. Okay. Versus gram-negative, which have this smaller, that's in between two membranes, right? The outer membrane and the inner membrane is where you'll get this, peptoglycan cell wall. All right. And if any of you have taken a microbiology class, you've probably done a gram stain. Okay. Which is a lab technique used to classify whether a bacteria is a gram-positive bacteria or a gram-negative bacteria. Okay. So this violet purple dye, right? When you subject your bacterial colony to the dye, gram-positive bacteria will retain the purple color, okay, because peptoglycan binds to that purple dye. Now, gram-negative bacteria will not retain that purple dye because they have an outer cell membrane that interferes with the uptake of the purple dye. Okay. So, la-di-da-di-da, whatever, purple bacteria, pink bacteria, who cares, right? Why do we care specifically about these two types of, these two major distinctive classes of bacteria? Does anybody know? Why do we care about gram-positive? Why do we care about gram-negative? Absolutely. Really good. This is incredibly important because most of our antibiotics that we've developed work on gram positive bacteria in some way, shape, or form. Like penicillin, like directly disrupts this peptoglycan layer, right? Thereby killing the bacteria. Gram negative bacteria, right? These ones, because they have this outer membrane, we've discovered much fewer antibiotics, and you need to use these much more powerful, general antibiotics that kill, both species. So whenever you get sick and they test you if you have some sort of infection, right, they'll look and see if it's gram positive or gram negative, and you hope it's gram positive because there's a lot more antibiotics and they are way more effective and way more specific towards these gram positive bacteria. Okay. So prokaryotes, right? For a lot of reasons, they're just like much simpler than eukaryotes. There's no membrane-bound organelles. Their cells can uptake things much easier. This means that a lot of molecular research and genetic engineering is done on prokaryotes, right? And so prokaryotes are a really, really, really good tool to study biosynthetic pathways, to study gene functions. It's easier to do it in bacteria, if it's something that's conserved, right, and test different pathways and this and that. Okay. And so you can use these, engineered auxotrophs, to select and to confirm, lots of different genetic and metabolic research. Okay. So the idea is that if you were to have a pathway in a bacteria and you were to knock out a critical enzyme, for in this case, if this is the pathway to create arginine, an essential amino acid, and you were to knock it out, then this bacteria all of a sudden can't grow unless arginine is fed artificially, right? So the only way for this thing to grow is, right, if you feed arginine, right, you insert a new gene, or set of genes that can bypass or replace the knockout. And 3, or the bacteria mutates / evolves on its own. And I used to work in a genetic engineering lab, specifically doing process engineering and fermentation. And I can tell you right now that you would be so surprised if we would create these strains of different bacteria and yeast, in fact, where we would knock out essential parts of their metabolism, that they can actually, over the course of, weeks, figure out how to overexpress different shunts and pathways that actually go around. And they can maybe not evolve, but evolve pretty quickly. Okay. So now the idea is, right, in this example, that if you have these bacteria with arginine knocked out, right, that it won't be able to grow on minimal media, right, where there's no arginine. And so the only way it will be able to grow is if you plate these bacteria on media where arginine is added. All right. And so just, I want to talk about this. This is just some nomenclature. If you ever see an enzyme or, a molecule and it's got a minus on it, that means that it's been knocked out or not present, versus something with a plus means that it's present. All right. Okay, let's talk about gene transfer for prokaryotic cells. The first class would be conjugation, right? Which is where, certain prokaryotic cells can actually take in DNA from the environment. Whether that be from, other lice bacteria, or if there's, a colony of bacteria signaling to each other, right? What you can get is actually this thing called a pilus, right? Which can actually extend out and make contact with a neighboring bacteria, in which case it can do this circular replication of a plasmid and replicate it and transfer it over to the new cell. Okay, so specific names that you need to know. If something is F plus, that means that it's the donor cell. F minus is the recipient cell, right? Meaning, again, plus present, so it has the plasmid present, and F minus meaning it doesn't have it, it's absent, okay? So now at the end, they both have it, so they're both F plus. Okay, there's something called transduction, and this is where genes can get transferred via bacteriophages or via viruses, where you can actually have a virus bind to a bacteria cell, inject its own DNA, right? Which can get in here, hijack the system, create a bunch more viruses, lyse the cell, viruses get out, right? Okay. And sometimes, the DNA can actually undergo homologous recombination and actually insert itself into the genome. All right. So that's transduction. And then my favorite one, transformation. This is where, this is where DNA can come from the environment, right? So like I said, other lice bacteria, or even better, molecular biologists, right, can actually introduce foreign DNA. And then, if you're in a lab, you've probably done this, an electroporator, which sends an electric shock. And so the DNA can actually uptake a plasmid or some sort of foreign DNA. And now you've created a transgene, right? You've inserted DNA into a bacteria host that can now replicate it and do everything, right? So very critical, very critical understanding that bacteria can uptake, a lot of DNA through transformation, right? So now you can express foreign DNA, you can do your genetic engineering, you can test different genetic cascades and so on and so forth. And you're going to be running into that a whole lot. All right. Okay, let's go over viruses. Last but not least. And technically, as of today, viruses are not alive, and that's because they cannot sustain themselves. The only way for them to propagate is that they need to infect hosts, okay? And so viruses are very cool because they can have a lot of different genomes, unlike anything else. They can have single-stranded DNA, right? They can have single-stranded RNA. They can have double-stranded RNA, double-stranded DNA. They have a lot of different options for how their, simple, small genome is comprised of. And there's two main infection methods. Yeah, that's. It's been a debate for a long time. I think our whole lives it's been that they're not alive, but obviously viruses and RNA is like the first molecules first, right? It's like what's alive, right? The fact that they're still replicating, and they're have infected how many people with COVID, right? Are they alive, right? So who knows? But as of today, they're technically not alive because they cannot sustain themselves. So we'll go over the lysogenic and the lytic life cycle, okay, which is either inserting themselves or into the host's actual genome and being dormant, or where they actually can lyse the cells to multiply, to spread. Okay. And so if you hear the word retrovirus, right, that just means that it's a RNA that has a positive sense RNA genome, right? And it carries a reverse transcriptase. These are the two cycles, right? The lytic cycle, which again, this, if a virus can infect a bacteria, it's called a bacteriophage, right? And they have these protein capsids, and then they have their genetic information within that capsid, and it can infect the cell, right? And once this, genetic information is in the cell, it can use the bacteria's, DNA polymerase, all the things that needed for DNA replication and for, protein synthesis can happen there. And then if it's in the lytic phase, it'll just keep multiplying and making new viruses to a point where the cell will burst, right? And the viruses can spread to wherever they are in their environment. And then there's, of course, the lysogenic cycle, right? Which is where you can get this homologous recombination, where this foreign DNA actually can insert itself into the genome and then just lie dormant there, right? And so, as your cells multiply, its gene multiplies, it's that new DNA multiplies too, right? So more and more cells will have it, right? And then, depending on certain events, that little genome can actually cross itself out and start to replicate again and replicate again and replicate again, and then go into the lytic cycle. All right. So that's pretty much it. That's, basic biology in a nutshell. But before we go, you have one more thing, if you want to stay. And that's another practice question. Oh, and thanks, Jonathan. Yeah, it's a long lecture. And I'm just going to say they're all going to be long. I mean, I'll try to shorten them up a little bit. It's just so hard. So hard. There's just so much to do. Yeah, you think this one's content heavy? Wait till, we have metabolism, which is like, metabolism is a lot of metabolic pathways. So this Sunday, it's actually technically not biochem. This Sunday, it's going to be, or Saturday, it's going to be physiology. Yeah, so that's going to be Spencer. And he's going to do all the physiology lectures, which is cool. Physiology is fun because a lot of people haven't done physiology, in school. So it's all new and it's all like, oh man, this makes sense. This is why, my body operates this way. Cool. Oh, did I not? Oh, I didn't even go to the last. Sorry. Sorry about that. Here's the last passage question. If you guys want a little extra practice right now. Oh, and someone says, well, we have a lecture about how to interpret lab experiments. Yeah, that'll be the last lecture, but also not the last lecture. You're going to get that in the cell bio lecture. You're going to get that in the genetic expression lecture, right? Molecular bio lecture. All those, use lab techniques and we'll present data in, graphs and tables similar to this passage that's actually just put up right now. This is very, very MCAT. This is, something like you would see on an MCAT, right? And so as we do these practice questions, right, it applies the concepts we learned, and how the AMC will present it. And so we'll actually go over it and what to look for, what not to look for, what all this technical jargon means. So yeah, if you guys want to, I can go over whenever you guys, I don't usually go over until someone throws a guess out. So we've got one for C. Another one for C. Another one for C. And I can go over question 7, which will probably help you answer question 8. Okay, I'll do that. That's probably a good idea. So this is classic, right? You come up to an MCAT passage, and now all of a sudden you got these, acronyms, OALH. You got these, names for enzymes. You got these metabolites. You've got, you've memorized the amino acids, but now you're like, what the hell is L-homoserine? What the hell is L? Everything just sounds slightly different, and it seems unfamiliar. Okay, that's going to happen to you, and you need to immediately not panic, right? And you need to immediately start to simplify your life, right? And just trying to figure out, what do you actually need to know, and what does everything mean, right? So for this passage, it's describing this bacteria, right? And it's saying that these two metabolites, SALM, which stands for S-adenosyl L-methionine, is necessary for survival, but also LHC, which is L-homocysteine, is necessary for survival. So in order to grow, it needs this and this, right? So those are the requirements, okay? Now, there's these 4, right, separate auxotrophs, right? So we want to find out, what is missing in these auxotrophs. What enzyme does this auxotroph not have, and, can it grow or not? Okay, so to do that, for each one of these, you feed minimal media, right? This just means, growth media with missing everything. And then you one by one add each of these metabolites in there. Okay, and then you can test whether it grows or not. Okay, so if we're looking at auxotroph B, right? I'm going to start to erase these so we can go over it. Right, auxotroph B likely has a deficiency in which of the following enzymes? Right, so here are the enzymes, right? And those are going to be this step, this step, this step, this step. Those are going to be the things that convert metabolite to the next metabolite, to the next metabolite, to the next metabolite. Okay, and if we're looking at auxotroph B, we really don't care about anything else, right? You can just cross out everything else, simplify your life, get rid of all this extra information that the MCAT presents you. Okay, and so it doesn't grow in minimal media. Okay, now you add this metabolite, OALH, and it still doesn't grow. So what does that mean? That means that this might be missing, this might be missing, and this might be missing, or some combination thereof, right? Because it can't convert this metabolite into these 2 end products that are needed for survival. Okay, so then we move over to the next one. MM plus LM, okay? And it does grow. So, right, we've moved on to LM, and you've added this, and the metabolite grows. Okay, what does that mean? It means that, let me erase this. It means that for sure, SAM1P, the cell has SAM1P because you go from LM to SALM, okay? But it also means that the metabolite has this and this, because this is also necessary for growth, okay? Which means that the only enzyme that it could be deficient in is MET15P, right? Does that make sense to you guys? Because I can explain that again and again and again. Yeah. Okay. So you need SOLM and LHC to grow. Okay. And so in order to do that, you have to process LHS down to these LAHS to LM to SALM. Let me get out my marker, right? You need to be able to keep moving this way, and you also need to move this way. Okay, and we need to find out which one of these things is deficient specifically for metabolite B. Okay, so the best thing to do is just systematically go one by one through your feeding experiment, right? And so here, if you feed it just OALH, just this, and it doesn't grow, that means that either this step is messed up, this step is messed up, or this step is messed up, or some combination of. Does that make sense? So we don't know which one, but we know that one of these must be messed up, right? Because it still doesn't grow. And we've already bypassed MET2, so we don't even care anymore if this is deficient or not, right? So then you go to the next step, okay? Right here. Minimal media plus LM. So now we're feeding LM. So we've bypassed this step, right? We've bypassed this. So is this thing present? Absolutely. So this does not have a deficiency because under LM it grows, right? So this must be produced. Okay. Does that make sense? Oh, and right here, right? What is it you said? I'm also interpreting the data as if it grows OHS, so MET2P works. Okay, so now let's keep moving down the line, right? We have to get to the end here. LHC, right? If you feed LHC, right, that's an end product, right? So if you feed LHC, you would expect it to grow unless one of these is messed up, okay? And then here, if you feed it SALM, you would expect it to grow unless one of these is messed up, okay? So based on, here, too much writing, right? If you look at SALM right here, right, that means I fed it this and it grows, which means that by default, this thing has to be working, right? Does that make sense? And so you can eliminate 16P. Start eliminating answers. Why does LHC work if SOLM works? Because it grew, right? In the passage right here, very good question. Both SOLM and LHC are necessary for survival. So that means if you're feeding this right here, if you're feeding SOLM, then somehow it has to be creating LHC. So you know that the MET16P has to be working, okay? Therefore, you can eliminate MET16P, okay? And so the strategy here would be to go through and find which ones must be working and start eliminating answers, okay? So where's the other condition where it grew? Probably a good place to start, right? LM, right? So we fed it this, okay? And it grew, right? That means that SAM1P must be working. So now we can eliminate SAM-MP. Does that make sense? And so let's go to the OALH, right? You fed it OALH, right? But it didn't grow, right? And we know SAM-MP must be working, right? Where's LHS? Do we feed it LHS? No, we never fed it LHS. So how do we know MET2P is not working? Yep, exactly. Cool. So do you guys see how this works? And so the answer here is B. Would that also automatically mean that M16 is working if LM alone shows growth? So if you have LM, yeah, that's exactly what I did the first time, right? If LM shows growth, then you know this has to be working and you know this has to be working. Great. Very tricky question. And this is very classic. Like, I mean, these knockouts, they have the knockout questions are classic. And then these auxotroph questions are classic, where they'll do a knockout and you have to decide, you know, what broke, right? And so here's another practice. Let's do question number 8. And this one's it's the exact same type of question, but it's just like one more step, right? You're instead of doing the enzyme, now you're doing you're actually feeding. So we got one for D. Oh, and the final answer for this one is MET 15P. So let's go through this, right? We got an A. Yeah, so this is like the same type of question, right? Let's read this. What combination of two substrates added to minimal media would allow auxotroph C to grow? Okay, so now we're looking at C. So that's the only thing we care about here, right? And so we have to find out which one of these, just like we did for question 7, isn't working, right? And so you can go through the same logic and find out which of these there's a quote unquote, deficiency in. And so if C, oops, right, can grow on minimal media plus OA LH, right, then it's MET2 that's deficient. Does that make sense? So now we know that there's a deficiency in this enzyme. So now we need to find out what combination of two substrates added to minimal media would allow auxotroph to grow. So if there's an answer that you can just skip, like this is all we need to grow, right? Does this show up anywhere? No, 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 right? Okay, so now we actually have to, figure this out, right? L-homoserine and L-methionine, right? So this was good, L-methionine, right? Feed here. Does that allow it to grow? I don't know. What about L-methionine and S-adenyl L-methionine? Right, so that would be this one and this one. Okay. But then how do we get to LHC? Yep, so nope. What about L-homoserine and L-homocysteine? Right, and L-homocysteine. right? Well, we know this one's deficient, so wouldn't that one work, right? And what about answer choice D? Let me erase all this, right? So this is the one that doesn't work. L-methionine and L-homocysteine. Yep, that would work. Got that? Does that make sense? Cool. Yeah, so that's, classic understanding metabolites, intermediates, what happens when you knock something out, stuff. And that's a wrap. Eight was easier than 7 because you knew how to do it, right? 7 is hard just because it was all new, right? You don't know what to look for. Yeah, no problem. Lots of stuff. Things are going to pick up real fast here in bio. And it's going to be very overwhelming, but you just have to keep trudging along. And you'll slowly start to speed up. Yeah, the questions in the next lecture are going to be pretty bad. They're going to be good, but it's good. You got to train hard, and because the MCAT's going to be just as bad, if not worse. Yeah, appreciate you guys. Wow, yeah, I've heard that too. The MCAT is like, it's cliche, but the MCAT is like the activation energy, right? Into medical school. Maybe the step 1 was harder, but at least you're already in the system. You're already phosphorylated your glucose. You're already stuck in the cell. And someone asked, is there a feature for you to turn on live transcript / closed caption? You mean like if it translates my words on the screen? I don't know. If there is, I don't know how I don't know. Yeah, thank all you guys. Great questions. You guys are on top of it. It's good. It's really good. The notes for this lecture, Marissa? Oh, I didn't email them out. I just sent, you mean the slides? I sent the slides to the chat. I'd have to make you notes from this lecture. Yeah. But there's also the 2 handouts that are on the portal pretty much cover everything in here, plus the section 6 of the first chapter of the bio book 2. That will cover everything. Yeah, of course, Tiffany.
Hi, can you hear me?
Yeah, totally.
So my